membrane structure, and within membrane structure looking at membrane lipids, specifically sphingolipids and glycolipids. So let us look at the learning outcomes of this session. Sphingolipids are structural components of the cell membrane, just like phospholipids are structural components of the cell membrane. Similarly, sphingolipids are structural components of the cell membrane, and they are metabolized to signaling molecules that lead to diverse functions. That means on getting broken down, sphingolipids are giving rise to certain molecules that can signal to carry out various diverse functions, which will include cell development to cell death. So all of these can be regulated by the signaling through the sphingolipids. Sphingolipids comprise sphingospholipids and glycosphingolipids, which both contribute to the asymmetry and dynamism of the membrane. Sphingolipids distribution in the membranes is diverse, and so is glycolipids, and they're able to interact with other membrane components to form structural and functional domains in the membrane. The distribution of glycolipids and the sugar type on the lipid influences functions of a cell and also contributes to forming a protective outer environment on the membrane. So let us one by one look at both sphingolipids and glycolipids and together how they contribute to the functioning of the membrane. So let us look at the basic structure of sphingolipids. So when you look at sphingolipids, the backbone of the sphingolipids is basically the sphingosine unit, where you can see that you have a single hydrophobic acyl chain, which with a group containing what is called as the NH2 group at one point, and here you have the OH group. Now, if this H is replaced by a phosphoric acid, as you can see over here, then it forms what is called as sphingosine 1-phosphate, and this then becomes the backbone to form the sphingomyelin. So the sphingomyelin, as you can see over here, is when the phosphoric acid that is attached to the choline, okay, then that is what is forming the sphingomyelin, and we know that many of the mammalian cells have a high content of sphingomyelin on the outer leaflet of the bilipid layer. So and secondly, when you have the OH replaced or substituted by when you have the NH2 in a bond, an amide linkage with the fatty acid chain, okay, then you have what is the simplest form of glycosphingolipid, and that is what is ceramide. Now when you have the H being substituted, say by for example a glucose moiety, then that is what is forming a glucosyl ceramide, and therefore this is what is glycosphingolipid. And this is the sphingophospholipid, which we also consider as a phospholipid in the membrane. Now, generally what has been observed is whether it is sphingophospholipid or whether it is glycosphingolipid, you would find that both the chains, this acyl chain as well as this acyl chain, both the chains are saturated. So you will not really find a kink or a bend in this because of the absence of cis double bonds. So this is something that is important to note. Then you have glycolipids. What are glycolipids? When lipids are bound to sugar moieties, then they are called as glycolipids. So for example, you can see over here how you have a lipid moiety which is bound to only galactose. So then this forms what is called as galactocerebroside. And when you have a lipid moiety bound to an oligosaccharide, then that is what is called as a ganglioside. And this is what is typically a GM1 ganglioside found in several neuronal cells of the body. And what is important to note over here is that many of these glycolipids have a special sugar moiety which is called as N-acetyl-neuraminic acid, N-acetyl-neuraminic acid, and this N-acetyl-neuraminic acid is also called as a sialic acid. Now, these are present generally on the surface of the membrane, which means we are looking at the fact that it is present on the outer leaf of the bilipid layer, not the cytosolic leaf of the bilipid layer, only on the outer leaf. So, you can see how it is contributing to the membrane asymmetry. You will find most of these glycolipids present on the outer side of the bilipid layer and hardly any or negligible on the inner side. So you can already see that therefore across, if you have a line of symmetry, you will not find symmetry because on one side you have the sugars, on the other side you do not have the sugars. Interesting also to notice that these sphingolipids or the glycolipids, they can interact with each other either through interactions between the sugars. So that is one way of interacting. And the second is that between the acyl chains as well, you can have interaction because of the van der Waals forces. So this is interesting because what has been observed is that because of them coming together and interacting with each other, they are able to form what is called as microdomains in the bilipid layer. Currently, also what has been observed is that many of the gangliosides, because of the presence of a negative charge on the sugars, are able to contribute to the membrane getting a net negative charge. So you can also see that the membrane gets slightly the potential across the membrane. There is a contribution not just because of the ions across the membrane, but you can also have a certain amount of charge on the membrane because of the presence of sialic acid, say for example, in many mammalian cells. So they contribute to giving a net charge to the surface of the membrane. Now this is important for two reasons. One, if you have a net negative charge, then it would repel anything that is also negative. So in a way, it can create an environment which is going to repel anything that is light charged. Like mentioned earlier, it is responsible for forming microdomains in the membranes, and we will look at it a little in detail now. So, as mentioned, it can create a very typical outer environment for interactions. So, suppose you have this membrane, and in this membrane, you can see how, along with the phospholipids, are present several other components, like, for example, the sphingomyelin, the ceramides. You have several of the sphingomyelin coming together to form a microdomain. So, you can see here how there's a collection of certain molecules, like for example sphingolipids, cholesterol, then you have certain proteins, integral membranes that can come together to form, and of course they're interacting with phospholipids, so they come together to form a certain domain in the membrane, and these domains are also called as the lipid rafts. Okay, so together they would move in the sea or ocean of phospholipids. So what is understood is that all of them, because they are interacting with each other, they are forming a small domain, and this small domain can actually be moving around as a raft, and therefore they are called as lipid rafts. Now please understand that these are transient. They do not exist for too long. They form, they assemble, they carry out a specific function, and then they can disassemble again. So that is something that has been observed. Now, what is also interesting to note is that because of all of these glycolipids and sugar and sphingolipids, especially glycosphingolipids, you can see that you have an array of different sugars present on the surface. And these array of different sugars present on the surface create a certain environment. So you can have this surface, this membrane interacting with its extracellular matrix and also with another cell in which is close or which is in proximity. So these things are important. Now, other thing that you can notice is that most of these sphingomyelin, glycolipids, etc., are formed by the endoplasmic reticulum. They go into the Golgi, and from Golgi they are then taken into the plasma membrane. So membrane. So basically what we can understand by this is that the synthesis of the glycolipids, the synthesis of the phospholipids, the synthesis of the sphingolipids all are generally through the endoplasmic reticulum, which then goes to the Golgi where it gets sorted, and from there it goes to the different membranes in the cell. So this is something that is very, very important. Now, certain glycolipids have also been found on organelle membranes. It's not just found on the plasma membrane, it can be also found in intracellular membranes. Now, they are mainly involved in signaling. Now, when we say signaling, what does it mean? It means that when these interact with the outside environment, they are able to get metabolized to form. So, for example, in this case, okay, you have an enzyme that is going to enable it to form what is called as a ceramide platform, which in turn is very specifically going to interact with something, and then there is a signaling responsible. So these signals that the uh, sphingolipids and the glycolipids can kind of form by forming another metabolized. So we say that you have glycolipid can be metabolized to give rise to a lipid messenger, and that lipid messenger will give a signal to another molecule that will give the signal to another molecule, and then you can have a cascade of signals, and that cascade of signals finally leads to some kind of a physiological response. So questions like whether a cell should go in for development further or whether a cell should die, even such questions are kind of addressed by the sphingolipids and the glycolipids that are present on the surface of the membrane. Now, many of these sphingolipids and glycolipids are also able to kind of give an identity to a cell type. Okay, so because it is present in this cell, the cell is, say, for example, cell X. If that is not present on the cell, then the cell may be cell Y. So, therefore, each cell type can get an identity because of the presence or absence of certain sphingolipids and glycolipids. But please do keep in mind that an identity of a cell is not dependent only on sphingolipids and glycolipids on its surface. You also have glycoproteins being present, and many of these glycoproteins can also be responsible for being a surface marker. So you can identify a cell because of the presence of a particular molecule, and the particular molecule may be a sphingolipid or a glycolipid. That is important to note. Now, because you have on the surface, therefore, an array of different sugars present, these sugars together form a kind of microenvironment, and this microenvironment is about nanometers distance from the actual membrane, and therefore, this kind of forms a protective barrier. So, all these sugars that are present on the surface could be because of a glycoprotein, could be because of glycolipids, could be because of proteoglycans, but all of them have sugars on their surface, and when you have these sugars on the surface, they contribute to membrane dynamics. So one of the things that this glycocalyx does is it forms a protective layer over and above the plasma membrane, and so this is what is called as a glycocalyx. Now, presence of a certain type of sugar can also be a kind of hotbed for viruses to interact, or they could be molecules to which the surface proteins on the viruses can bind. So whether the pathogen can invade a particular cell type or not would depend very much on what is the glycocalyx that is present on the surface. Secondly, I told you that you have many such molecules that are present on the surface, and they become a kind of identity for the cell. So many immune cells which are moving around in our body, when they see these molecules present on the surface of the cell, they would recognize that cell as self, and therefore not do any harm to the host or the normal cell. Okay, say, let's say, for example, take the example of sialic acid. Now, sialic acid, that is N-acetylnuraminic acid, which is present in several glycolipids. These sialic acids are present in most mammalian cells, but they are not present on microbes. So, therefore, our immune cells will recognize the microbes as foreign particle because they do not detect sialic acid on the surface, but a normal cell, okay, could be recognized as self because it has identified the presence of the N-acetylnuraminic acid present in there. So there are molecules present on the immune cell that can interact with the sugars present on the surface and determine whether the cell is self or the cell is non-self. Based on that, it will either phagocytose or call in neutrophils, macrophages, etc. to take care of the non-self, or if it is self, then it is not going to initiate the process of an immune response towards one's own cell. So that is again another important facet. Now, another important fact that facet of glycocalyx is in signaling itself. Because it has this n number of sugars present on the surface, they can interact with other molecules on other cells or molecules present in the extracellular matrix, and that interaction can lead to a signal transduction, which can then finally decide what response the cell is going to give to that signal. So signaling is one important function attributed to sugars present on the surface. So in this session, we are definitely looking at sphingolipids and glycolipids, but what you can also observe very clearly is that we are also talking about membrane carbohydrates, because sugars in the membranes are either associated with proteins, or they are associated with sphingolipids, or they are associated with lipids. So in the form of glycolipids. So therefore, the glycolipids and the sphingolipids contribute to the glycocalyx or contribute to the membrane carbohydrates per se. Now we look at the lipid rafts. We just looked at previously, and we talked about something called microdomains. So microdomains are also what is called as lipid rafts. So as you can see over here, in a membrane, what can happen is several components of the bilipid layer can come together to interact with each other. So say for example, you have sphingolipids coming together, as you can see. You can have the sphingolipid holding a transmembrane protein. You can have a sphingolipid attached to a GPI anchored peripheral protein, or you can have it bound to a cytoplasmic protein. Now, all of these coming together and interacting with each other is forming a domain within the membrane. So you can see that this part does not have any lipid, has any microdomain. It is devoid of any specific domain. They are all just phospholipids that are in constant motion. Okay, in this case, there may be motion, but then all of them are moving together. Okay, so they're all hand in hand, glove in glove, working together to carry out a particular function, probably. Okay, so these lipid rafts are definitely not permanent structures. They're only assembled or formed for a particular action, and once that particular activity is carried out, then they can disassemble again. So therefore, it is difficult to study the lipid rafts because they are only present for a transient time, only for some specific moments, and then they kind of again disassemble. But that is something very important, and there are many such microdomains formed in the membrane. They are formed, they are disassembled, formed, disassembled again and again. So you can see how dynamic, how many changes happen in the membrane all the time to ensure some function or the other. Now, lipid rafts are typically, therefore, an assembly of phospholipids, sphingolipids, cholesterol, and GPI anchored proteins. So, whenever one looks at what is lipid rafts made up of, then you would find that these are the components which are most commonly found in lipid rafts. And interestingly so, because they are able to interact with each other with greater affinity, which is why they come together, which is why they assemble together also. And interestingly also, cholesterol, sphingolipids, and phospholipids, they are able to kind of enable change in the conformation of the proteins that it is associated with, and thereby it can affect a particular function. So let us make the conclusions. Sphingospholipids and glycosphingolipids comprise the sphingosine backbone with esterified fatty acids. Most glycosphingolipids are present in the outer leaflet of the bilipid layer, with the sugar moiety facing the extracellular environment. These are able to form a protective and interactive glycocalyx. The sphingolipids are able to interact with each other and other components to form microdomains or lipid rafts to carry out specific functions. There are a lot of studies that have shown direct involvement of sphingolipids in cell signaling and immune responses. Thus, sphingolipids and glycolipids contribute to the asymmetry of the bilipid layer and confer special physical properties to the cell to carry out specific functions. They also contribute, therefore, to the dynamism of the membrane. Thank you.